everyone, it's Dr. Marcon, and this lecture is on mitosis, the cell cycle, and cancer. So this goes along with chapter 6 in your book on reproduction at the cellular level. So we're going to answer some questions with regards to this topic. What does cancer have to do with mitosis, and how did Henrietta Lacks contribute to our understanding of cancer? So our learning objectives include distinguishing between mitosis and meiosis, identifying the different stages of mitosis and cytokinesis, as well as identifying causes of cancer and how it affects the cell cycle. So here are some other guiding questions to get you through this lecture video. Why do cells divide? How do cells divide? What controls cell division, and how is the process different in cancer cells? So here we see a picture of normal cell division, and then we can see a picture of cancer cell division. Now, are cells constantly dividing or only dividing at certain times in our lives? Or maybe both? Now, how was cancer treated prior to World War II? Most physicians would either zap it or cut it out, not with scissors, of course, but with proper surgical tools. But for the most part, we'd either do ablation or some sort of surgical procedure to cut out any cancer that we could see. Now, strangely enough, mustard gas was used in World War II, and it actually led to the discovery of chemotherapy drugs. But how do chemotherapy drugs stop cancer? Now, we know that most chemotherapy drugs do interfere with an important part of a cell's life cycle, and this is cell division. Now, we need cell division, of course. Many cells must reproduce throughout our lives, and this helps us with the healing process. We have to have red blood cells constantly made so that we can have transportation of oxygen. Cells are important for our immune responses, growth and development of the human body. So we can see, as we age, we grow and we develop into adults, and we need constant cell division to allow that to happen. So we know that cell division helps replace old and damaged cells, helps with the repair of our body. As we can see in this picture, a little girl has a little scab or wound on her knee, and we need those cells to help replace the damaged cells, creating a nice little scar right there. And again, cell division allows for growth and development of the body. So we know that cells have different lifespans. Now, can you guess how often these cells divide? We know that we have different types of cells. We have red blood cells, we have cells in the stomach, skin cells, liver cells, muscle cells, as well as nerve cells. However, each cell has different lifespans. For example, the red blood cell. We know that red blood cells are produced every 120 days because the lifespan of the red blood cell is 120 days. Stomach cells. Stomach cells are replaced every day because we constantly use these cells, especially when we have production of acid in our stomach. Also, cells that protect the lining of our stomach are constantly worn away. So we need stomach cells to be constantly dividing and replacing old cells. Skin cells. We know that skin cells divide every day, and we have a new skin each month. So we're constantly shedding off skin cells. Liver cells. Liver cells, not so often. So liver cells divide about every year. And then muscle cells rarely divide. They just get bigger. Nerve cells, again, nerve cells rarely divide after birth. So we have a limited number of nerve cells. But before we learn about cell division, we have to learn some of the terminology. Sexually reproducing organisms, such as humans, have two types of cells. We have somatic cells, also known as body cells. Examples of this include our red blood cells, our platelets, also known as thrombocytes, and our leukocytes, or our white blood cells. And then we have our reproductive cells, or sex cells, and these are known as gametes. So the two types of reproductive cells that we have include an egg or ova and the sperm cells. Now, there's a distinct difference between somatic cells and gametes, several actually. So we know that the majority of the cells in our body are our somatic cells, and we only have two examples of our reproductive cells or sex cells. Also, the number of chromosomes. So somatic cells have 46 chromosomes, or 2 matched sets of chromosomes. The letter N, for example, when we start describing the number of chromosomes or sets of chromosomes, the letter N is used to represent a single set of chromosomes. So since somatic cells have 2 sets of chromosomes, they are considered diploid and are designated as 2N. Okay, so let's draw that. Oops. So somatic or body cells are considered 2N, okay, N representing a single set of chromosomes. So 2N equals 46 chromosomes, all in all. Whereas gametes only have 1 set of chromosomes. So these sex cells are considered as haploid. Haploid meaning 1 set, and are designated as N. Okay, so 1 set, 46 divided by 2 is 23. Okay. So make sure you know the difference between diploid. So 2N is diploid, which equals 46, and N equals 23, which is considered haploid. So diploid for 2N, diploid. And then haploid for N, haploid. Okay. So again, reproductive cells include sperm, which is produced by a male, and then an egg, which is produced by the female. Another difference between somatic cells and reproductive cells is that our somatic cells or body cells divide by a process known as mitosis, whereas our reproductive cells divide by a process called meiosis. Now, DNA, we know, is found in all cells. DNA is a molecule of heredity, provides the instructions for building an individual organism. So a cell's complete complement of DNA is called its genome. In eukaryotes, the genome comprises of several double-stranded linear DNA molecules bound with proteins to form complexes called chromosomes. We know that, and we talked about monomers of our important macromolecules in our body, DNA being one of them. The monomer of DNA are nucleotides. And we know that nucleotides are made up of a phosphate group, a 5-carbon sugar, as well as a nitrogenous base. Now in DNA, we know that the 5-carbon sugar is our deoxyribose sugar. And the nitrogenous base will determine if the nucleotide is a pyrimidine or a purine. The pyrimidines include thymine and cytosine, and the purines include adenine and guanine. Okay, so these are the bases that are found in the nucleotides of DNA. And a way to remember which ones are pyrimidines and which ones are purines, just remember the mnemonic pure as gold. So purines include adenine and guanine. Now we know that there are greater than 3 billion of these nucleotides in human DNA. So 3 billion, greater than 3 billion adenine, thymine, cytosine, and guanines. And when a cell divides, it must organize the DNA into chromosomes. And we talked about chromosomes earlier. Chromosomes are a single large DNA molecule tightly wrapped around proteins. So these are complexes of DNA molecules wrapped around proteins. So how many chromosomes do you have in your body cells? How many chromosomes do you have in your gametes? So we talked about this earlier. The number of chromosomes are different in body cells versus gametes. Again, we use N to equal the number of unique chromosomes. In our somatic cells or body cells, they are considered diploid or 2N. So we have 2 sets of chromosomes. So 2 times 23 equals 46 chromosomes. Okay, so 1 set will come from mom and 1 set will come from dad. With gametes, which are our sex cells, such as our egg and sperm, our gametes are considered haploid. So N, again, equals the number of unique chromosomes. N, just by itself, is equal to 23. So 1 set of chromosomes. Okay, so again, body cells or somatic cells are diploid. 2 sets of chromosomes, which equals 46. Haploid, only 1 set of chromosomes, which equals 23. And again, number of unique chromosomes. So we use what is called a karyotype to allow us to visualize the chromosomes in a cell. So this is a picture of what the karyotype would look like. And the chromosomes are numbered 1 through 22, and then that last pair being our sex chromosomes, determining whether or not we are male or female. So this is what a normal karyotype looks like. And we can see that we have homologous pairs of chromosomes. So 1 set we get from mom and the other from dad, and together they make a pair. Now, homologous chromosomes are the same length and have specific nucleotide segments called genes. And we know that genes are the functional units of chromosomes that will determine specific characteristics, such as eye color, such as hair length, such as ear size, and they determine these specific characteristics by coding for specific proteins. We know that the last pair are the sex chromosomes, X and Y, and they're the single exception to the rule of homologous chromosomes. So they're not the same, and they determine whether or not a person is male or female. So XY means that the person is male, and then XX will determine that the person is female. So again, homologous pairs of chromosomes, each one within a pair carries genes controlling the same inherited trait. So when body cells or somatic cells divide, they do so through the process of mitosis. They will have the same number of chromosomes as the parent cell. So we know that the parent cell is 2N, 2 sets of chromosomes, for a total of 46 chromosomes. So through mitosis, the parent cell will duplicate that DNA and then place that DNA in 2 daughter cells that have the same number of chromosomes as the parent cell. So here's that parent cell. And here are the 2 daughter cells that will have the same number. Again, will be diploid, 2N, which equals 46. So we talked about, so now we're going to talk about how do cells divide. Now, are they constantly dividing, or are they only dividing at certain times in our lives? And what exactly controls cell division? With regards to cancer, cells that divide out of control cause cancer. So here we see some normal cells in pink, and then we see cancer cells growing through the normal tissue. So basically your cell's gone bad. And here is a link. So I actually just checked out this link, and I changed the link so that it actually works. Now this link shows how tumors grow, showing abnormal cell growth, and then they undergo a process called angiogenesis. This is when there's a formation of blood vessels that will give blood supply and nutrients to the tumor, allowing for this abnormal cell growth. What occurs afterwards is metastasis. So metastasis is the spread of the tumor to other areas and organs of the body. So kind of wreaking havoc all over the body. Then we are going to talk about the cell cycle. The cell cycle is an orderly sequence of events leading to cell division. And we want to think of the cycle like a clock, with events occurring at different times within the phase of a cell's life. So we have actually a really good video here on the cell cycle that you guys can watch. And we also have within the cell cycle certain checkpoints, but we'll talk more about these checkpoints later. So the first phase, of course, of the cell cycle is interphase. Now this is where the cell stays 90% of the time. And we have different phases within interphase. First, we have the G1 phase, or the GAP1 phase. Then we have the S phase, which is known as the synthesis phase. And then we have G2, which is GAP2 phase. And then once interphase is finished, we then go into mitosis. And mitosis is basically the division of the nucleus within a cell, along with the division of the DNA from the parent cell to the daughter cells. And then finally we have cytokinesis. So cyto referring to the division of the cytoplasm of the cell. So we can see that the majority of the cell spends most of its time in interphase, about 90%, and then goes through the mitotic phase, or M phase, which takes up about 10% of the cell's cycle. Now I know cats often ponder, where do dividing cells spend the majority of their time? And as we can see, the majority of the time is spent in interphase. So first we'll talk about the G1 phase, or the GAP1 phase. At this point, this is that first growth stage. We have production of more organelles, more proteins, and phospholipids. So just going over some of the phases within the cell cycle. So we know that the cell life cycle is a series of changes a cell will undergo. And that first phase, where the majority of the time the cells they stay, is interphase. And G1 phase, again, growth 1, or GAP1 phase, other ways that we call it, is that first part of interphase. At this phase, the cells are metabolically active. They make proteins and they grow rapidly. And this phase is very variable in length. It can occur from hours to even years. So, for example, our sex cells, or egg cells, can stay in this phase for years. At this point, we have structures known as centrioles that begin to replicate near the end of G1. And centrioles will be important, and we'll talk more about centrioles when we get to the mitotic phase. So then we have, after the G1 phase, the synthesis phase. This is when we have doubling of our chromosomes. What does a duplicated chromosome look like? So again, chromosomes will duplicate during the synthesis phase, or the S phase. So here we have our chromosomes, which will then duplicate into sister chromatids. Okay, so we go from one DNA molecule to two DNA molecules. Okay, so from single chromosome to sister chromatids. Then we have the G2, or GAP2 phase. Again, just more growing, more of a growth stage again. So just a bit of a summary of those two phases. So again, S is the synthesis phase. This is where DNA will replicate itself. That ensures that daughter cells will receive identical copies of the genetic material, where we have chromatin that is extended. And then we have the G2 phase, or the growth 2 or gap 2 phase. This is when we have the centrioles finish copying themselves, as well as enzymes that are needed for cell division are synthesized. Now, during our synthetic or synthesis and G2 phases, the cell will carry on its normal metabolic activities. So once we have finished interphase, the cell will then enter the mitotic phase or M phase. So the mitosis phase or M phase, this is the part of the cell cycle when the cell divides, specifically the nucleus. Now this occurs at the appropriate time where copied chromosomes will split and the cell divides. Now, are the resulting daughter cells identical? And the answer is yes, they are identical and they are copies of the parent cell. So a way to remember the different phases of mitosis is PMAT. So we have prophase, metaphase, anaphase, and telophase. Okay, so prophase is that first step in mitosis. So chromatin condenses to form chromosomes. The nuclear membrane will dissolve and the DNA will leave the nucleus. A mitotic spindle will form. This is a structure that will separate the sister chromatids. So prophase is known as the first and longest stage of mitosis, and it can be divided into early and late prophase. Early prophase, this is when we have the chromatin threads condensed into chromosomes. We know that chromosomes are made up of two threads called chromatids or sister chromatids. The chromatids are held together by a structure called the centromere. Okay, so if you actually draw out a chromosome, it has that specific kind of X shape. So we have one here and then one here, and held together by a structure called a centromere. Okay. Then we have structures called centrioles. Again, these centrioles are in pairs that separate from one another, forming that mitotic spindle that will kind of pull the chromatids apart eventually. In the late prophase, the centrioles will continue moving away from each other, and we have fragmentation of the nuclear membrane. Again, basically the nuclear membrane will dissolve so that the DNA could leave the nucleus. Again, centrioles are structures that will help organize that mitotic spindle. Before we get into the other phases of mitosis, we know that pharmacologically speaking, our cancer drugs are used to interrupt certain processes within the cell cycle so that we can prevent abnormal cell growth. One of these drugs is called TAXOL. TAXOL is a chemotherapy drug that will interfere with that mitotic spindle formation, which we know that occurs or begins to occur in prophase. TAXOL is a drug that is used to treat breast and ovarian cancers. About 1.3 million pounds of the bark of the Pacific yew is used to treat all women with ovarian cancer, which comes into question. Do we want to save a life by killing a tree? This was actually a huge topic of debate within the 1960s, back in the 1900s. And we know that in 1993, this was the first year that this drug came on the market. So again, coming from the bark of the Pacific yew, also known as Taxus brevifolia, to make that drug TAXOL. The next phase in mitosis is metaphase. This is the second step in mitosis. At this point, the duplicated chromosomes will attach to a mitotic spindle. So here we see those centrioles at the opposite ends of the cell, creating this spindle or spindle fibers that will attach to the chromosomes. So they form a single line in the middle. So when you think of metaphase, think of that mitotic spindle in the middle. And of course, alignment is important because we want to make sure that we have equal pairing of our chromosomes that will eventually separate and go to each end of the cell. The next phase in mitosis is our anaphase. This is the third step. So here, the sister chromatids of each chromosome will begin to separate and move away to the opposite ends of the cell. So notice how A is underlined, away is underlined. So this is when we have pulling apart of the sister chromatids away to opposite ends of the cell. Our fourth and last step in mitosis is telophase. So at this point, we have two brand new nuclei forming, and then the chromosomes will appear as chromatin, and we have disintegration of those spindles that were used to separate the sister chromatids, and this marks the end of mitosis. So mitosis, basically the duplication of DNA from a parent cell and a formation of two distinct nuclei. So after mitosis, we then have a process called cytokinesis. Cytokinesis occurs after mitosis. Basically, the cytoplasm is divided between the two new cells. The cell membrane moves inward to create two daughter cells, each with its own nucleus with identical chromosomes. So these next two slides show just a summary of the cell cycle, starting with interphase and then going through the various stages of mitosis and then ending in cytokinesis. So that first phase of the cell cycle where the cell stays about 90% of the time, we have interphase. Interphase, this is the phase or period of a cell's life when it carries out its normal metabolic activities. Again, interphase is not part of mitosis. And we have the three distinct periods of interphase. We have G1, S, and G2. Okay, so then we start with mitosis. And mitosis is composed of prophase, metaphase, and anaphase. Prophase can be divided in early and late phases. So in early prophase, we have the chromatin coils and condensation forming our chromosomes. Okay, so we can see the chromosome consisting of two sister chromatids that are attached at a structure called the centromere. And then we have formation of an early mitotic spindle with the centrioles starting to kind of move to the opposite ends of the cell. So by late prophase, we have breaking up of the nuclear envelope. We can see it dissolving, allowing for the spindle to interact with the chromosomes, starting to kind of pull them apart. Okay, so we can see those centrioles at the opposite ends of the cell starting to kind of pull apart those chromosomes. We then enter metaphase, and again we see that metaphase plate lining up in the middle of the cell. So the two centrioles are at opposite ends of the cell. We have the lining up of the chromosomes, and then with that metaphase plate. And then when we go to anaphase, we have the daughter chromosomes being pulled apart to the opposite ends of the cell. So anaphase is the shortest phase of mitosis. So the chromosomes split simultaneously, and each chromatid will now become a chromosome in its own right. And then finally, the last stage of mitosis is telophase. This begins as soon as movement of the chromosomes stop, and you have basically reformation of that nuclear envelope. And then we have, so the nucleus is forming, and then we have basically a contractile ring occurring between the two cells. It's called a cleavage furrow. And at this point, we have the beginning of cytokinesis, where we have the pulling apart and formation of two distinct cytoplasms. Okay, so that's basically a good summary of the different stages of the cell cycle that I've explained. If you'd like, you can actually go to this link to find a good video on the different stages of mitosis. Okay, so this is the end of part 1 of the lecture video. The part 2 will focus on cancer and how cancer affects different stages of the cell cycle.